Hello students, welcome back to a new video of Sunanda's tutorial. Today we will discuss the whole chapter of photosynthesis in one shot. So let's start. First of all, what is photosynthesis? Photosynthesis means synthesis of food or photosynthete using the sunlight. And the main producer of the food chain is the green plants because they have chloroplast and chlorophyll pigment in it, which can capture solar energy and helps in the preparation of food for the plants. Photosynthesis is a physico-chemical process by which green plants use light energy or radiant energy to form or produce or manufacture the organic compounds. Photosynthesis is an anabolic process. That means something is forming in that process. Something is produced. And endergonic process, that means energy capturing process, and a redox reaction, that means reduction and oxidation both occur in photosynthesis. Water is oxidized to form O2 while CO2 is reduced to form sugar. That means water is oxidized. Splitting of water occur in photosynthesis and it oxidized to form oxygen, which releases into the environment, while CO2 carbon dioxide is reduced. What is the meaning of reduction? Reduction means the addition of hydrogen to form sugar. So basically photosynthesis is a redox reaction because both reduction and oxidation occur in photosynthesis. Oxidation of water molecule occur to form O2 or oxygen to the environment. Reduction of CO2 occur to form sugar. Then what is the importance or significance of photosynthesis? It is the primary source of food on earth. It is the only process that liberates oxygen that we inhale. Photosynthesis convert light energy to chemical energy, and that chemical energy is stored in food that we eat. So it is an energy transduction reaction. All energy released by burning of coal, wood, natural gas directly or indirectly captured from sunlight by photosynthesis. Then historical background, history of photosynthesis. Aristotle and Theophrastus both believed that plants absorb food from soil. Von Helmont, he worked on willow plant and concluded that weight of the plant increased due to water. Then John Woodward said that beside water, soil also increased the weight of the plant. Then Stephen Hales concluded that green plants obtain food from leaves. Joseph Priestley, who did a complete experiment on the photosynthesis using the green plants, he proved that green plants clean air and proved his concept using a bell jar experiment. So bell jar experiment was carried out by Joseph Priestley. He observed that a candle burning in closed space bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in closed space. He concluded that a burning candle or an animal that breathed in air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that mouse stayed alive because green plant produce oxygen. But at that time, it was not discovered that oxygen is produced from the green plants. But he observed that when he put a mint plant inside that closed bell jar, the mouse stayed alive and candle continued to burn. Priestley hypothesized that plant restore to air whatever breathing animal and burning candles remove. Simple. Next, Ingenhouse discovered that green plants purify noxious air in the presence of light. Then D. Saussure showed the significance of water in photosynthesis. Drottruket cleared that the importance of chlorophyll pigment in photosynthesis. Sachs showed the product of photosynthesis is starch. Blackman proposed the law of limiting factor. What is limiting factor? Law of limiting factor tells that when a process is controlled by many factors, the process is actually limited by the pace of a slowest factor. Then Engelmann plotted the action spectrum of photosynthesis. Warburg found that the unicellular algae, chlorella, as a suitable material to study photosynthesis. Then Van Niel proved that sulfur bacteria use S2S, that is hydrogen sulfide, in place of water as an hydrogen and electron donor in photosynthesis. Then Robert Hill demonstrated the photolysis of water. During photosynthesis, photolysis of water occurs, that is splitting of water molecule. Then Ruben, Kamen and Hosseth proved that O2 given out or released during photosynthesis comes from water, and they used oxygen 18, that is isotope of oxygen they used, and proved that the oxygen released during photosynthesis actually come from the water. Then Arnon discovered the process of photophosphorylation. What is phosphorylation? Phosphorylation means formation of ATP, and photophosphorylation means when the ATP is released in presence of sunlight. Then Kelvin discovered the path of C3 cycle, which we will discuss in details later. Then Emerson discovered two pigment system. Hatch and Slack discovered C4 pathway. So let us discuss this photosynthesis process in details. Photosynthesis occur in many bacteria, and bacteria also have chlorophyll. They have a specific type of chlorophyll which is known as bacterial chlorophyll. Then algae also can perform photosynthesis. Green plants perform photosynthesis obviously. Sometimes generally this photosynthesis process occur in the green leaves, but sometimes it occur in stem part also. Roots of Trypantinospora plant also perform photosynthesis, so they are known as assimilatory roots. Chloroplast or green plastid are the cell organelle where photosynthesis occur in the plant cell. Inside the plant cell, many organelles are there. Plastid, a green plastid is present in green plants, which is known as chloroplast. There the photosynthesis processes occur. Then within the leaves, mesophyll cells have large number of chloroplasts. So mainly the leaves perform photosynthesis. It is the structure or diagram of the chloroplast. It is the outer membrane, then inner membrane, then stroma. Stroma is the liquid part of the chloroplast. Then grana. Grana are the stalk of thylakoids. Then ribosomes. Many ribosomes are present. Some starch and lipid granules as reserve food material present inside the chloroplast. So these are some photosynthetic pigments found in the plants, and among these all, chlorophyll A is the main pigment. Chlorophyll A is the main chlorophyll pigment found in all green plants and mainly perform the process of photosynthesis. Chlorophyll has a tadpole-like structure with porphyrin head and phytol tail. Head is a tetrapyrrole or 4-pyrrole ring structure with a central non-ionic magnesium atom. So magnesium atom is present in the chlorophyll. Chlorophyll A has methyl group and B has aldehyde group. So remember these three important points as it generally coming in your competitive exams. Then phytol tail is a long hydrocarbon tail attached to the porphyrin head. Then absorption spectrum and action spectrum. So please read these two terms thoroughly as these are very very important. What is the difference between absorption spectrum and action spectrum? See, the visible spectrum of light wavelength between 390 to 700 nanometer is used for photosynthesis, is known as photosynthetically active radiation. Then the red light is most effective for photosynthesis. In red light, the photosynthesis rate is maximum, followed by blue light and the green light where the least photosynthesis occur. Absorption spectrum is a graph. showing the amount of different light wavelength absorbed by a pigment. And action spectrum is a graph that shows relative rate of photosynthesis in terms of oxygen release at different wavelength of light. Then what is quantum? Light ray consists of tiny particles. A light ray consists of tiny particles known as photons, and the energy carried by photon is called quantum. The number of photons or quantum required to release one molecule of oxygen is known as quantum requirement. On the other hand, number of oxygen molecule released per photon of light is called quantum yield. Then what is red drop effect and Emerson enhancement effect? At wavelength beyond 700 nanometer, it was observed that apparently insufficient energy to drive any part of photosynthesis. So huge drop of in efficiency has been noticed at 700 nanometer. This phenomenon is called red drop effect. A slight drop in the rate of photosynthesis was observed after or beyond 700 nanometer, and that was known as red drop effect, and it was noticed by Emerson. Later Emerson applied two wavelength of light, one red light, one far red light, and simultaneously, and he observed that the rate of photosynthesis enhanced, and that is known as Emerson enhancement effect. Then organization of photosystem, pigments in photosystem. Inside the chloroplast, grana, that is stack of thylakoids are present. In that thylakoids, the photosynthesis occur on the thylakoid membrane of chloroplast. The pigments are organized in thylakoid membrane in two discrete photosynthetic unit called as photosystem 1 and photosystem 2. Photosystem 1 is located in non-appressed part and 2 is located in appressed part of the thylakoid. Photosystem concept was first given by Emerson. So one photosystem consists of a reaction center, which is the main chlorophyll A pigment, and a light harvesting complex. And light harvesting complex consists of an antenna complex and many proteins. So what is the mechanism of photosynthesis? Photosynthesis is a physico-chemical process where the light energy is converted to chemical energy, and this process occurs in two steps. One is light reaction, which is directly dependent on the light, and another one is dark reaction, which depend on the products of light reaction. So let us discuss about light reaction. Light reaction follows this process. The first step is light absorption, and then transfer of electron, then photolysis of water, that means splitting of water and release of oxygen, then formation of assimilatory power like ATP and NADPH. Then what is photophosphorylation? Photophosphorylation is the formation of ATP in presence of sunlight, and it is of two types. One is cyclic, the second one is non-cyclic. In light reaction, cyclic photophosphorylation occur where the electron released from photosystem 1 moves a series of coenzyme and return back to the same photosystem 1, which is known as cyclic electron transport or cyclic photophosphorylation. Cyclic electron transport involves only pigment system 1. This situation is created when activity of photosystem 2 is blocked. When there is lack of photosystem 2 or it is blocked, only photosystem 1 will work and cyclic photophosphorylation occur. Under this condition, only pigment system 1 is active and photolysis of water does not take place. Blockage of non-cyclic ATP formation, and this causes a drop in CO2 assimilation during dark reaction. There is a consequent shortage of oxidized NADP. So what will occur in cyclic phosphorylation? P700 molecule, which is PS1 or photosystem 1, excite a electron which is first taken by ferredoxin FRS, then plastoquinone, then cytochrome BF complex, then plastocyanin, then returns back to photosystem 1. During this electron transport at two places, ATP formation occur. So at last, during this cyclic photophosphorylation, two ATPs are released. Then non-cyclic photophosphorylation. In case of cyclic photophosphorylation, the released electron returns back to the same photosystem 1, but here there is no return of electron. The electron released from PS2 goes to a series of enzymes and coenzyme and photosystem 1. This is called non-cyclic in electron transport chain or Z scheme, as it looks like a Z like manner. The synthesis of ATP in non-cyclic transport is called non-cyclic photophosphorylation. What is the main function of this non-cyclic photophosphorylation? To produce assimilatory power like NADPS2 and ATP. But in case of cyclic photophosphorylation, only ATPs are formed. No NADPS2 formation occur in cyclic photophosphorylation. But in case of non-cyclic photophosphorylation, both assimilatory powers, that is ATP and NADPS2, released. Photophosphorylation process results in the movement of electron in a non-cyclic manner. For synthesizing ATP, using the energy from electrons, excited electron provided by photosystem 2 is called non-cyclic photophosphorylation. This process is referred to as non-cyclic because the lost electron by P62 of PS2 are occupied by P700 of PS1 and not revert to P680. So it is not like your cyclic photophosphorylation. It is the overall non-cyclic photophosphorylation where the splitting of water takes place at first, which provides electron to PS2. After the absorption of sunlight, the water molecule splits to form hydrogen, oxygen and electron. This electron is captured by the PS2, which moves to quinone, then plastoquinone receive it, then cytochrome BF complex, then plastocyanin. Then this electron is transferred to the PS1, which will received by the ferredoxin reducing system. Then it will transfer the electron to ferredoxin. Then NADP capture this electron and with the help of H plus they form NADPS2. And during this movement of electron, ATPs are always produced at the time when it passes through the cytochrome BF complex. So at last NADPS2 and ATPs are formed. Next, photolysis of water. The process here I have mentioned that photolysis of water occurs first. The splitting of water molecule in presence of light is called photolysis of water. It occurs in the luminal side of thylakoid, that means lumen side, stromal side. So it occurs in presence of manganese, chloride and calcium. So it all known as oxygen evolving complex, and the process was first discovered by Robert Hill, so known as Hill's reaction. Then chemiosmotic hypothesis. Peter Michael postulated that the chemiosmotic hypothesis, it explains the mechanism of ATP synthesis within chloroplast during photosynthesis. During photochemical phase or light reaction, ATP and NADP are generated. These are key components and used in dark reaction for the production of final product of photosynthesis, that is sugar molecule. As per the chemiosmotic hypothesis, ATP production is the outcome of proton gradient which is established across the membrane of thylakoid. The required component for chemiosmosis are proton gradient or proton pump and ATP synthase. ATP synthase is an enzyme bringing about ATP synthesis. The enzyme has two portions, one F0, one F1. F0 is on the transmembrane channel and F1 activate the enzymes. They phosphorylate ADP, that is adenosine diphosphate, to form adenosine triphosphate. ADP or adenosine diphosphate phosphorylate to form ATP, that is adenosine triphosphate. One of the driving force of ATP synthesis is the protein gradient that is developed across the membrane. So here, when the light reaction or the non-cyclic photophosphorylation occur, during that process, a proton gradient is formed. So that is the main factor which provides or which helps in the production of ATP. During photosynthetic electron transport, hydrogen protons accumulate inside the thylakoid. Hydrogen protons are released into thylakoid space after each split of water molecule, because water molecule splits here and hydrogen accumulated inside the thylakoid. So as a result, H plus ion concentration increased inside the thylakoid than the outside of the thylakoid membrane. Then as a result, H plus released outside to maintain, to balance the H plus concentration, and they release through a ATP synthase. And during their release, ADP is phosphorylated to form ATP. So it is the overall reaction of chemiosmotic hypothesis or overall mechanism of chemiosmotic hypothesis. Then the dark reaction. ATP and NADPH2 produced during the light reaction. It is finally consumed or consumed and help to perform the dark reaction. It is light independent phase, but it consumes ATP and NADPH2 and cause the CO2 fixation or reduction to form glucose or sugars. It is a series of enzyme catalyzed reaction, and it occur in stroma part of the chloroplast. Light reaction occur in thylakoid membrane, but it occur in the stroma part of the chloroplast. CO2 fixation is the second phase of photosynthesis and explained by 3 cycles. One is C3 cycle, one is C4 cycle, one is CAM cycle. In some plants, the first product of this C3 or dark reaction is a 3 carbon compound, and that plants are known as C3 plants, and they perform C3 cycle or Kelvin cycle, as this whole cycle was discovered by Kelvin, Benson and Bessam. This cycle have 3 phases. One is carboxylation, where the initial carbon fixation occur. The second phase is reduction or glycolytic reversal, where one sucrose or starch formed, sugar molecule formed, then regeneration of RUBP. In case of Kelvin cycle, the first acceptor of CO2 is ribulose 1,5-biphosphate, which accepts CO2 to form phosphoglyceraldehyde, which is a 3 carbon compound, and the enzyme which help in this process, first phase, is known as Rubisco, RUBP carboxylase oxygenase. So then in some plants, the first product of this dark phase is not a 3 carbon compound. They are the first stable product of this dark reaction is a 4 carbon compound, and the first reaction is aided by the, not by the Rubisco. Here in C4 it is aided by the pepcase or phosphoenol pyruvate carboxylase, and the first receiver of CO2 in C4 plants is PEP or phosphoenol pyruvic acid. Phosphoenol pyruvic acid absorbs the CO2 to form oxaloacetic acid, which is a 4 carbon compound. Then it will be converted to malic acid. Then malic acid converted will move to the bundle sheath cell, where the actual Kelvin cycle occur. Then malic acid splits in bundle sheath cell to pyruvic acid, and one CO2 is released, which will be accepted again by the RUBP, which is present in bundle seed cell, and pyruvic acid transported back to mesophyll cell, where it is converted to again phosphoenol pyruvic acid. In case of C4 plants, they have two types of cell in their leaves. One is mesophyll cell, one is bundle seed cell. In mesophyll cell, only PEP carboxylase is present, but in bundle seed cell, normal rubisco is present. So initial carbon fixation is aided by the pepcase or PEP carboxylase, and the first stable enzyme, first stable product is oxaloacetic acid. So next is another type of cycle which was performed by the CAM or crassulacean acid metabolism cycle, performed by the arid plant or desert plant, where the plant this condition is adopted by the arid plant or the desert plant, where photosynthesis during the day occur but only exchange gases at night, where in this case, during the day, night time, the stomata open, and during the day stomata close to prevent the water loss, because this type of cycle occur in arid condition. So during dark time or in absence of sunlight, stomata open, CO2 absorption occur. First acceptor is PEP, which convert it to oxaloacetic acid, then it will be converted to malic acid. It is stored in the vacuole. In daytime, malic acid again splits to form pyruvic acid and a CO2 accepted by the RUBP and perform the Kelvin cycle, and in this way this desert plant carry out the photosynthesis by crassulacean acid metabolism pathway or CAM pathway. Then what is photorespiration? Some C3 plants, in C3 plants, the first enzyme which helps the carbon fixation is called as the rubisco, which is a dual natured enzyme, that is carboxylase and oxygenase. Sometimes instead of carbon dioxide or low carbon dioxide concentration, they receive or absorb O2 or oxygen and perform photorespiration, which is a highly wasteful process, because during photosynthesis ATP is released, energy is released, but here in photorespiration two extra ATPs are consumed. This process of photorespiration occur in three organelles. That means it requires three organelles to complete the whole cycle of photorespiration. One is chloroplast, second one is peroxisome, third one is mitochondria. So it is the whole cycle of photorespiration, where the first rubisco accept the oxygen and convert it to phosphoglycolate. Then phosphoglycolate is formed the glycolate. Then glycolate moves to peroxisome, where it is converted to glyoxylate and aminated to form glycine. Then glycine moves to mitochondria, where it is converted to serine by releasing CO2 and NH3. Then serine moves to peroxisome again, where it is converted to hydroxypyruvate. Hydroxypyruvate again form the glycerate. Glycerate moves back to the chloroplast, where it form the phosphoglyceric acid, and this phosphoglyceric acid again moves to this Kelvin cycle, and normal Kelvin cycle will be performed. So it is a highly wasteful process. So then the factors affecting photosynthesis. What are the factors that affecting photosynthesis? One is light. Light is the source of photosynthesis, and it is the most important raw material of photosynthesis. The effect of light on photosynthesis can be studied under these three headings. One is light intensity. Under low light intensity, rate of photosynthesis is low. Increase in light intensity causes increase in the photosynthesis rate. Then light quality. Photosynthetic pigment absorb visible part of the radiation. For example, chlorophyll absorb blue and red light. Usually plants show high rate of photosynthesis in blue and red light and least photosynthesis in green light. Then duration of light, longer duration of light period favors photosynthesis. Generally, if plant get 10 to 12 hours of light per day, it favors a good photosynthesis. And the generally the photo period that a plant require to flower. Then carbon dioxide, if we increase the amount of carbon dioxide in lab condition, and if the light and temperature are not limiting factor, the rate of photosynthesis increases. This increase is observed up to 1% of concentration, but the CO2 concentration beyond 1% is highly toxic to the plant. Then temperature, the rate of photosynthesis increase in the temperature up to 40 degrees Celsius, but beyond that the rate of photosynthesis decreased, because in high temperature the enzymes gets degraded, so the process of photosynthesis stops. Similarly, in very low condition, the enzymes get inactivated and the process of photosynthesis stops. So the optimum temperature for photosynthesis is 35 to 45 degrees Celsius. Next, water. Water is a essential raw material for photosynthesis. Then oxygen, high amount of oxygen or high concentration of oxygen is shows the inhibitory effect on photosynthesis. It is called the Warburg effect. Then some mineral elements are required for photosynthesis, which are magnesium, iron, copper, chlorine, manganese, phosphorus, etc. There are some other external factors which regulates the process of photosynthesis. So these are the factors which affects the process of photosynthesis. Next, the most important term, CO2 compensation point. What is CO2 compensation point? The point reached in a plant when the rate of photosynthesis is equal to rate of respiration. This means that carbon dioxide released from respiration is equivalent to which is taken up during photosynthesis. This compensation point is reached at as light intensity increases. So it is the point where rate of photosynthesis is equal to rate of respiration. So it is all about your photosynthesis. I hope you really enjoyed the session, and I hope that you will get benefit from it. Thank you.